So our next segment will contain two speakers speaking about data in astrophysics as well as data in genomics. The first speaker will be speaking about the Open Data Challenge in computational astrophysics. Dr. Daisuke Nagai is an associate professor of physics and astronomy at Yale University and the faculty co-director of the Yale Center for Research Computing. His current research focuses on theoretical and computational modeling of the structure formation of the universe and its application to cosmology. He obtained his PhD from the University of Chicago in 2005. He was the Sherman Fairchild postdoctoral scholar at the California Institute of Technology before he joined the Yale faculty in 2008. Welcome, Dr. Nagai.
Thank you very much. Okay, great. Well, thank you very much for the invitation. It's a pleasure to actually talk about some of the perspectives that we're actually learning from computational astrophysics area. So I would like to start, I think this is a really exciting time in science where we might be actually going through some sort of paradigm shift in science. If you look back in the history, the science has been, over the long time, the thousands of years, it's mostly the empirical studies of describing natural phenomena by collecting data. There was a paradigm shift several hundred years ago when the theoretical branches arose, where Isaac Newton actually started to use the mathematical formulations to actually describe laws of nature and tried to, in the hope of describing the empirical data that the astronomers were collecting. And that opened up that theoretical branch several hundred years ago. And since then, we've seen the emergence of this field of physics, which have highlighted identify several laws of physics from electromagnetism to gravitational physics to quantum theory, which has been teaching us a lot about the physical laws of nature. But in the last few decades, we have actually seen another emergence of new technique. This is the computational branch. And then today, due to the emergence of computations that actually starting to produce the large amount of data, that maybe that might be a new paradigm shift, that maybe the data is actually telling us something, and this new shift actually requires new ways of thinking to do science. The computation permeates throughout the society, starting from all the way from the smallest scales of particle physics all the way up to the structure formation of the universe, and everything happens in between, from molecular dynamics and application to drug discovery to looking for fusion research and in the hopes of actually finding clean energy. Car safety these days, in order to study the safety of automobile, we do no longer actually crash the car, but we do computational simulation to getting insights into how to actually build a safer car. If we want to actually train a pilot, we would actually use the flight simulator to actually train the pilot instead of having them actually fly on the actual physical plane, and so forth. And on the much larger scale, the weather forecast, planet formation, galaxy formation, and large-scale structure formation, these are the phenomena that's very difficult to actually study, and that the computational approaches has been really revolutionizing the way we actually understand those systems. So the computational power, in using computation, what we're actually doing is to actually trying to give a complete evolution of the physical system where you have a parameter that you can vary, and we are starting to do the experiment using computers. And this is sort of the new way of actually doing science. And that as the computer gets faster and faster, the data is growing with exponential rate. And with that larger data, it's even starting to change whether how we actually think about the science. Typically, we develop a hypothesis, go out and collect the data to test the hypothesis. But however, when you have so much data, you can start to think about whether the data can actually teach us something about that we don't know. Could the data teach us more about the ideas that we could generate? Those are possibilities that were and potential that is actually making this field particularly exciting. Data is becoming increasingly open and public. and that the fact that the data is everywhere is actually bringing people from physical sciences to life sciences to medical sciences together, connected through statistics and computing. So we might actually be in this rare opportunity where we can actually exploit this paradigm shift, and what comes out is, I think, is what we're here to do, to actually discuss. How can we actually make this field the most fruitful as possible? In my area of astrophysics has been inherently and traditionally data-driven, because we cannot do experiments on the universe that we are interested in knowing. So typically, so what's on the left-hand side is the Planck satellite mission that's in space, observing the universe in microwave. What's on the right-hand side is Sloan Digital Sky Survey, studying the galaxies' distributions in optical. And I sort of think of this as a cosmic genome project, where we are actually mapping out the distribution of mass in the entire universe. In the microwave, you can study about the distribution of mass in the very early phase of the universe, where the universe was very smooth. And what's shown on the right-hand side, the pictures of distributions of galaxies in the entire sky, is actually showing the universe is very clumpy, very homogeneous. There are regions with a very high density concentrations of galaxies, and there are regions with the empty voids. And so we have a snapshot of how the universe evolved from the very smooth state to very clumpy state. But in this field, we also know the physical law that's governing the evolutions of this physical system. On the largest possible scale, the gravity is the dominant force, and we have a working model of the gravitational laws developed by Einstein. So here what we're doing is we're taking the map that we see in the early universe from the cosmic microwave background as an initial condition. You can then take that, set that initial condition and solve the Einstein's equation forward in time, and you should be able to reproduce the distribution of a clumpy universe in the present day universe. So this is what has been done. And it actually generates the universe that consists of the gravitationally bound objects like galaxies and clusters of galaxies, and also there are empty regions like voids, as we see in the data. So what we're really doing, the modern astrophysics is really the nexus of the big data and supercomputing. On one side, we have a telescope as an instrument, which is taking a snapshot of at any given point in space. And on that, but however, on the left-hand side, we have the high-performance computer. It also acts as an instrument. In fact, we do most of the experiment where we vary the knobs in using these high-performance computers. Both of these instruments generate huge amount of data, which has to be analyzed through the data analysis pipeline. You use statistics to actually compute the relevant statistical quantity. You glue those information together through the statistical framework like Markov chain Monte Carlo, and then trying to infer things like what the universe actually consists of. Today, we believe that the universe that best fits the observation actually consists of the 4% of stuff that we knew in the last about these are matters, protons and electrons, and it turns out that this 96% of the stuff that fills the universe is made out of things that we didn't know about. This field of studying the universe in this kind of way has really taught us that all the physics that we knew in last until the last century only made up 4%, really opening up a new avenue for discovery. What's the nature of dark matter? What's the nature of dark energy? What is the universe made of, and where we're going? Now, to do this, on the left-hand side, on the computational modeling, there are different types of code that are available to actually model the physical evolutions of the universe. And here I'm showing the 6 different types of code. All of these are solving the same set of equations, but using different techniques. Some are using representing the distribution of mass as a particle. Some represent the distribution of mass as a grid and cells. And you solve the same set of equations in the different ways. And so in order to have a credibility, to actually test how the code is actually working, you try to use the different types of code and solve the same problem. This is a simple problem where you have a gas blob moving in a tenuous medium. and use the 5 different code to actually study to solve the exact same problem. And that the interesting thing, the problem here is that depending on what kind of code or algorithm that you use, the evolution of this physical system produce different results. So this is sort of the reproducibility crisis that we face in astrophysics, that the computational results do depend on code and algorithm. So if for any given algorithm, you can reproduce the same results, but although you're solving the same problem, if you're solving them using different technique, it could result in different results. Here is a slightly more complicated situation where we're using the 12 different types of code. So this leads to, for the computational astrophysics, what's important is to do this thing called the code comparison project. You need to develop different types of approaches for solving same problem, and starting with the exact same initial condition, trying to solve a slightly more complicated problem, in this case, the formation of galaxy cluster, the most massive objects in the universe. So here's an attempt back in 1999 where the 12 simulators got together, decided to simulate the same thing since they were not agreeing among themselves on the results that they were getting on the same problem. And you can see that, well, this is the distributions of gas in this massive galaxy clusters. And you can see that more or less that they actually form something. They're a spherical blob, but there are clearly some differences too. What's more notorious here, here's I'm plotting these distributions of gas as a function of radius. And there are the results actually agree fairly well at the very large radii, but there are notable differences in inner region depending, again, on the code. Whether you use grid-based or particle-based code, you get systematically different results. So this kind of comparison can actually teach us about the pros and cons of different techniques. So the scientific results, especially computational results, must be not only reproducible, but it would also have to be accurate. This also raises a question as to how do we actually know that we're converging on that right answer, which is also important as well. Okay. Now the reproduces challenges. So far I talked about how to run simulation and using different types of code, you get different results. The computational astrophysics involve fairly complex workflow where you have to go through, take the simulation outputs, visualize them, analyze through many, many layers of steps. First, identify the object that you are interested in, how you define the object. How do you actually analyze the data? There are fairly complicated algorithms that go through to process those outputs of the simulations and to go through the analysis pipeline, finally plot the results of the interested quantity. So the complex workflow, we also need to identify the different types and levels of reproducibility and accuracy in this research process in order to get the robust results. In this process, most of the intellectual contributions and assets actually are encoded in that code, which consists of 2 things: algorithm that we use to actually to develop a software, and the software itself. So the code is where the most of the intellectual contributions are, and then there's also the additional contribution lies in the workflow of the code, that interconnect between different types of code at different stages of the research. Some of the solutions are specific to research community, such as algorithm and software. However, there are also common solutions. Those include software sharing and preservation, which we use GitHub and Bitbucket to actually do that software development as a team, also with the scientists around the world. In terms of data sharing, we use Globus Online extensively to disseminate the outputs of the simulations and share them around with scientists around the world. The data science policy is another area where there are some commonalities among the researchers who do research on different disciplines. Those include the e-journals, funding agency, infrastructure, are some of the things that we share. Even though you're trying to actually solve the different problem, there are things that we can actually coordinate. So the Yale Center for Research and Computing can actually address some of these issues that are common among the researchers. And those include the cyber infrastructure such as HPC and storage, software, education and training, and grant support and community outreach, including the Day of Data, and organizing and bringing researchers together from the different disciplines to actually talk about the data or things that are common. In this way, we can improve the efficiency and actually exchange the different techniques that are used and different ideas. And I think this is the way forward in terms of making the transformative impact in different fields. So let me just conclude by saying that both reproducibility and accuracy are important for credibility of e-science. Open data and open data, open code are certainly the important solution. In our computational astrophysics, we are also learning that the algorithm and software development is equally important aspect of doing the science in this area. And we can use combination of those as a basis for data-driven science, hopefully in the future. Some of the solutions are specific to the research community or even sometimes to the group. Code development, process and workflow. In this area, the discipline-specific data scientists can actually play a very important role in developing and optimizing some of these software development and also the efficiency of the process and workflow. Some of the other solutions that are more common among the different research communities include HPC infrastructure, software, and data and code sharing. And those are the things that the Yale Center for Research and Computing will be able to help. However, there's some missing gap that I notice as we actually develop, trying to figure out what kind of research should be done within our own group, what kind of things should we actually coordinate and do it as a part of the community. Another important thing that we are starting to realize in our research is the algorithm development. As you develop a code, as I highlighted, the computational results could actually depend on not just what problem you're solving, but how you're solving that problem. And that sometimes we sometimes do not know. Basically, the problem that we are facing is that you're solving the same set of equations, and it's well tested by solving the problem that we know the answers to. But we're taking that code and extrapolating it to the regime where we don't know how well that code actually performs. Different types of algorithm has pros and cons. So when you actually extrapolate the code into take it into the regime where we haven't tested, but this is the whole point of doing the computational modeling, is that opens up a solutions to some of the problem that has not been solved before. But it's actually difficult to convince ourselves. How do we know that we're actually converging to the true answer? Given the code, you can reproduce the results. But if you use different code, you actually find two different results. We don't. In that case of the galaxy cluster, where we see differences in the grid-based and particle-based code, there's a long debate about which one is actually the right solution. We just get the two different solutions, and we don't know which one is the correct solution. The problem is very serious, because the comparison I showed two comparisons. Those are separated by 15 years. We're still debating about the same problem after 15 years of research, and this has to do with and the only way we can move forward is the independent development of different types of code, different types of algorithm, and then the community has to think about, have to convince themselves a better understanding of pros and cons, the characterization of the code algorithm is actually the way forward in actually getting the true answer from this, from computational astrophysics. And this is important because we're using the computational models to infer something important results, such as the composition and origins of the universe. And if we're using the wrong models to actually interpret the data where we cannot do the experiment, we will be inferring the wrong results. So that so this is one area that I think we would need to actually make a real impact. We need not just it's important to actually make the data and code available, but it's also important to think about how do we actually these issues of the accuracy and how we actually convince ourselves the results we're getting is the right answer. So let me stop here, and thank you very much.
Thank you, Dr. Nagai. We will have Q&A for both speakers of this segment at the end of this second speaker. Our next speaker, Arif Harmanchi, will be discussing the quantification of private information leakage from genomic datasets. Dr. Harmanchi received his PhD from the University of Rochester in 2011. He joined the Gerstein Lab at Yale as a postdoctoral researcher in 2011 as well, and since then has been working on the development of analytical tools for bioinformatics research. His primary focus is on functional genomics data analysis.
Can we copy this? We didn't put it in yet. Yeah, sorry. Oh, man. Yes. Either PPTX or PDF, they're the same. Yep. Great, thank you. Okay. Thanks. Okay. Yep. Yes. Hi, everyone. It's a great pleasure to be here. I want to thank the organizers for giving me this opportunity. So I'm going to switch gears a little bit, and I'm going to talk about computational privacy. It may get a little bit technical, but I'll try to get it as easy as possible, get through it as easy as possible. So yeah, the title is Quantification of Sensitive Information Leakage in Phenotype Data. So let me try to give you guys a brief context here. So so privacy is obviously it's very important. And as we do more open science and we share more data, one problem that is going to come up is a lot of different data sets that contain sensitive information or personal information that they can be linked together. And if you think about an adversarial person, he may go and fetch those data sets and, well, he can invade privacy. So, so I guess one thing that we will need to think about in next several years is going to be how we can quantify the risks and what's the leakage, what's the sensitive information leakage in these data sets. I mean, it's not a solved problem at all. We need to think about it really carefully. And actually, NIH wants to push data sharing a lot more in the next several years. And actually, yesterday, I don't know if you guys follow it, this 21st Century Cures Act passed through the House, and actually that gives NIH the incentives to actually make researchers put their data in the central data commons that NIH is actually planning to produce in a couple of years. So these are going to be real challenges that we will face in a couple of years. And I would want to start with this example that we like to present. There's nothing to do with genomics. So there was this Netflix prize challenge that this Netflix company, I'm sure you guys know about it. It's the movie rental company that where these guys, they wanted to improve their movie recommendation systems, and what they did was from the huge database of movie ratings, they extracted this training data set, which consisted of these users. Well, these are real people. Well, their names are obviously de-anonymized. And there were these movies and what this guy rated this movie, when did this guy rate this movie, and this movie's rating. So higher is better, lower is worse. And the idea was the researchers would take this training data set, Buell models, machine learning models, and they would then test these models on another testing data set where the rating is taken out so that Netflix can go and cross-validate and see who wins. And the prize of this challenge was $1 million, so it was a pretty serious thing. And it went on for like 3 years, I think. And let me talk about this data set a little bit. So there are 100 million ratings for 500,000 users. On average, each user rated 200 movies, and each movie was rated around by 5,000 individuals. And it turns out this was not a very good idea, because these researchers, Narayanan and Shmatikov, they actually showed that you can take this Netflix data set, which was anonymized, and you can actually go to another completely different movie data set, the IMDb, and you can actually link these, and using pretty straightforward techniques, I won't go into the details of those, and you can actually identify who a lot of the people in Netflix data set, their identities and taken from the IMDb. So that was a big problem, and actually Netflix got sued, and they had to pay a pretty big price for this. And well, there are 4 basic things to note here. So the reason why this attack happened firstly was these are very big data sets. So if you think about it, if somebody has an account on Netflix, there's a very good likelihood that he or she is going to have an account on IMDb. And a lot of the users, they rate a movie. For example, you watch a movie, then you would rate it around the same time, and you would give similar ratings. And these researchers actually statistically showed that. Well, actually, they also did the actual attack. But they showed that statistically you can link any very big data set with a lot of features to each other. So it wasn't a very good idea after all. Okay, going into this phenotypic problem, phenotype-genotype linking problem that we have, the way we are thinking about this is, we have a lot of phenotype data sets. These can be, your electronic health records, or it can be, a lot of molecular phenotypes like gene expression levels, metabolomics, proteomics, and any omics that you can think about. And there are these genotype data sets which you might have gotten in 23andMe or some ancestry website or your hospital. These are going to get very, very relevant to our health system. And what's going to happen in our what we are thinking is going to be very relevant to what an adversary does, is linking these data sets, because recently, I think 23andMe, they announced that they genotyped their 1 millionth customer, so they have millions of people in their database, and there's a very high likelihood that the guy who gets their genotypes measured in 23andMe, they're going to go and do it in Ancestry too, because Ancestry.com too, because a lot of people like doing this stuff. But the 23andMe and Ancestry.com don't know about each other. So if a hacker goes and hacks these 2 data sets, there's a very good likelihood that he can go and link them with another data set that he gets from a hospital or somewhere. So so in our case, what's happening is we have the phenotype data set, the genotype data set. These are independently served. So the genotype data sets, we are assuming it's hacked, and phenotype data set is anonymized. We have these just the IDs and a very sensitive information, for example, HIV status, and a lot of phenotypic measurements. So these phenotypic measurements are some numbers. It can be your gene expressions, it can be your height, it can be your blood glucose level, or some other measurement. And these are variants. So 0, 1, 2 means your reference allele, or heterozygous, or homozygous alternate. So don't worry about those. But these are basically 3 valued levels of your genotypes at certain positions in your genome. And there are these public data sets called phenotype-genotype correlation data sets. So what's given here is a phenotype is correlated to a variant. So for example, like a position in your genome changes and your eye color changes. So if I look at your eye color screen, I have a very good idea of what your variant in that position is going to be. So, and then what we are saying is you can actually do a genotype prediction using these correlation data sets, because I can go from this phenotype measurement to this variant. But this step is stochastic, so there's going to be some errors that you're making when you're getting this. I don't know if you guys can see the laser pointer, the predicted variant genotype. So there's going to be a lot of errors here. But the errors actually don't, the attacker doesn't care about the errors that he makes in the genotypes. He's trying to link this data set to this one. So what happens is he does genotype comparison between these 2 data sets, and matches the genotype IDs to phenotype IDs. So genotype IDs are actually the identifying information, and he gets the referring to the genotype ID, the HIV status. So at the end of the day, he linked this genotype ID to this HIV status, and that's where the damage is done. So I will focus on these. So what I'm going to do is I'm going to kind of do an example of how easily this attack can be done, and I'm going to concentrate on these things called expression quantitative trait loci. So that's a long way of saying, you know, the genotype affects at expression level. So here again, like this, we have on the x-axis, we have the genotype is 0, 1, 2. As this value increases, or as you go from the reference allele to the alternate allele, your expression level changes. So if you, I mean, you can think about this like, you know, you have AA, AC, and CC values here. So as you change from one allele to the other one, your gene expression level increases. So a variant changes the gene expression level of a gene. And so what we want to do is we want to quantify how well you can predict. Like I said before, there's a stochasticity in this, you know, the prediction process, the genotype prediction process. So we want to quantify the predictability to the leakage. So for the predictability, what we are looking at is we have the gene expression level. How well can you predict that from the how well can you predict the genotype from the given gene expression level? So we connect this to this thing called entropy. Well, you don't have to think about it that technically, but basically higher entropy corresponds to, you know, more randomness, and it corresponds to lower predictability, and vice versa for low predictability. So at the end of the day, what we are trying to do is we want to come up with a metric for predictability. That is because there's this stochasticity in this linkage process. And then for this information leakage, we're basically asking, let's say we predicted this genotype correctly, and how much information does it give me? And this is basically this is like the surprisal of this of finding this genotype within the population, and we formulate this with this logarithm of 1 over the frequency in population. So don't worry about this formula. What it's basically saying is when you have higher genotype frequency, for example, if everybody has certain genotype at the variant in the population, it wouldn't really identify anybody, right? For example, I mean, if I take the brown-eyed people here, it wouldn't, I mean, if I knew a person is brown-eyed, I wouldn't really identify anybody. But if I knew that one person is green-eyed, and it's usually green eye, you know, is much less frequent, then you can actually, you know, identify that person more reliably. And again, low genotype frequency corresponds to a high information content. Okay, so actually, if you take the, you know, some data from this 1000 Genome Project and do these computations for the real, you know, EQTLs, these EQTLs meaning the, you know, the variants that associate with, correlate with the genotype, the gene expression levels, you know, the leakage and predictability is much higher compared to a real, to a shuffled set. So the, you know, the metrics that we're proposing to use are actually kind of meaningful. As you can see, they're much more spread along the diagonal. And actually, if you can overlay these allele frequencies, meaning like how much, like what is the allele frequency of that variant within the population, then you will see that actually the stuff that is identifying and that is useful to an adversary, that is the stuff that gives you highest leakage and that is predictable, is going to have moderate allele frequencies. And they, I mean, as expected, they have high correlations with the gene expression levels. Well, having said that, I mean, it's obvious that an adversary wouldn't use. So these were plotted. The previous plots were shown for just single EQTL. So he would actually use multiple EQTL. So this is where actually the trade-off comes up. So as you can see, like as we start from the best EQTL, on the x-axis we have the predictability, on the y-axis we have the leakage. The best EQTL can be predicted pretty well at 78%, but it gives you very small leakage. As you go up in the, you know, in your EQTL list, you know, top 2 EQTLs, it gives you, it's like 50% predictable, gives you more leakage, and at 5 EQTLs you have 20% predictability and like, you know, 7 bits of leakage. But as you can see, as the, you know, an adversary, what he would do is, you know, start taking these guys, start using like 5 EQTLs, but he would although he's gaining more information to identify individuals, he's actually losing predictability. So this is where there is a trade-off between how well you can predict these genotypes and how well you can identify individuals because of the stochasticity. So now moving forward to how you would really implement this in practice, we actually proposed this three-step of a linking attack, 3 steps of a linking attack. So the thing is, so you have a person. What we want to do is we want to select these phenotypes that we will use for attack. And then next thing is, for the selected phenotypes, you do a genotype prediction, and then taking the predicted genotypes and using some auxiliary information, you can go and link these, and finally estimate the reliability of how well you did linking. So now I'm going to talk about this extremity-based genotype prediction, but the idea is actually pretty straightforward. So what we're trying to do is I take, for example, my gene expression levels, right? So they're like my height. For example, if I have, let's say we have a variant, right, that correlates with height. So if I'm very tall or if I'm very short, then you can actually say, I mean, these are the extremes of that phenotype, right? Then you could actually, you would have a very good idea of what genotype I have, because as I showed before, the EQTL, these QTLs actually, like for example, if I'm on top here, I'm most likely in this genotype, right? Or if I'm here, like in the other extreme, I'm probably 0 for this variance genotype. I mean, this is similar to like if you're driving a very expensive car, you're most likely making a lot of money, right? But if you're driving something, like a not very expensive, you could be rich, or you may have just middle income. You may be rich, but you choose not to drive a very expensive car. I mean, this is a very similar analogy here. So what we are basically proposing is, if you're at the extremes of these distributions, you basically assign the extremes of the genotype. So you're either rich or you're very poor, depending on the car model you're driving. So when you use this genotype prediction, or if you use this genotype prediction to do this linking attack, it actually turns out. So don't worry about this. So this is basically how we select the EQTLs that we are using in the attack. And on the y-axis, we have the fraction of vulnerable individuals. So what this is saying is, if you use all the EQTLs, you actually get a pretty good linking accuracy. So this is for 200 individuals, and we did a simulation of 100,000 individuals, and accuracy drops a little bit, but still it's very close to 100%. And actually, if you had access to that person's gender or where he or she is from, like population, or both at the same time, because gender is pretty easily predictable from gene expression levels. So and then you get actually some improvement. If you look at here, it's a very marginal improvement, but it's very high already. But at these levels, there's actually a significant increase in the accuracy. So I mean, like I said, although this is getting a little bit technical, what we are showing here is you can do these linking attacks with a very simple approach. Well, you could basically actually ask, okay, this is like 70% accuracy if I choose this, cutoff for selecting EQTLs, but which 70% is, like, for example, I predicted 10 individuals, 7 of them are right, but only the person who has the actual phenotypes knows those 7 people. Is there a way to attacker to really identify those people? So actually, there's a way to do that. So there's a very simple metric we call this first distance gap. So so for example, in our example, we have 200 individuals. So I go and link them to the 200 individuals, or all the individuals like I have in the genotype data set. And what I do is, for each individual, I have 200 comparisons, right? So what I do is I sort those 200 comparisons with respect to this genotype distance, and I take the difference between second one and the first one. And it turns out if you use this metric to select these individuals, you can get a pretty high accuracy. Here, on the x-axis, we have sensitivity. That is how many of the individuals in the phenotype data set are correctly predicted. On the y-axis, how many of the people that he predicted are correctly predicted. So So depending on this D12 threshold, he can actually select a very small number to link. And then it turns out you can see up to like 40% of the individuals, you can get 100% accuracy for this. And if he used the whole data set, he would get 70%. But for 40% of the individuals, you can actually get 100% accuracy. So this is like 100 individuals out of 200 data set. This is not a small number. And then these magenta colors are actually showing randomly selected thresholds. You can see they pretty fastly converge to the 70% accuracy. And finally, this actually doesn't affect only you, but your relatives too, because your relatives have very high correlation in your genotypes. That is, I take part in, for example, in genotyping service of a company, and I do it for another, I get phenotype in another company. They link me, and then, but the linking that they do, the genotype data set doesn't have me, but my brother, for example, right? Then actually it would put him at risk. So that's what it's showing. So when you do linking, actually it turns out a majority of the individuals that score in the second rank are the relatives of the individual. So actually when you take part in a genotype or a phenotype study, you're actually putting your relatives at risk, which actually creates a big problems about how you define consent and all these I guess, complicated issues. So this is the end of my presentation. It was a little bit technical, I guess, but if you guys have any questions, I can try to answer them.
In fact, for the questions, I'd like to call Dr. Nagai up to the front, as well as Arif, so we can ask the questions of the two segment speakers.
Yeah. Sure.
Daisuke, astrophysics has always been at the high end of the data generation and computational scale. Do you see the field starting to skimp back on resources, or are they always going to be the most Pac-Man-like in terms of aggressively going after whatever's available?
I think I'm not seeing that scaling back in a way that, I think that the physics and astronomy has often and traditionally has actually pushed the resources to actually grow. For example, the Large Hadron Collider is probably the biggest producers in terms of the amount of data that they generate, is just sort of redefining and it actually has pushed the infrastructure that way. I think astronomy continue to do that. The kinds of large-scale survey that are coming online, like LSST, which is an optical survey to map out the distributions of galaxies in the universe, to SKA, which is a square kilometer array in radio. And there are several dozens of such survey coming online only to produce ever more exponentially larger amount of data per survey. So this is going to grow, and it's going to push the envelope of the production of big data, and it's going to define in terms of how to deal with that amount of data, how to actually how to build the network, how to do the storage. I think my guess is that I think astronomy will continue to actually experiment at this sort of frontier of big data and produce different types of experiment and I think will help also other field to sort of trying to produce the best practices from such experiments. So I'm not seeing the astrophysics dialing back down. In fact, I see more growth in terms of the amount of data that will be produced from astrophysics community.
So if I understood your talk correctly, I mean, one of the main messages is that you may think you're anonymizing your data when really there are many ways that people can re-extract that information. And so I wonder, with all these initiatives like All Trials, where they're really pushing getting all these clinical trial data out there, are they kind of working into these protocols a link attack susceptibility test? I mean, this seems like it should be a standard part of what they're doing, because it may not be obvious to them how they're putting those patients at risk. And do you know if there are any kind of movements to push that forward?
That's right. So, okay, so let me say something first before answering your question. When I do this presentation, people usually think like, we are terrified. Okay, so what do we do about it? But the idea is not really that. It's totally the opposite. So the thing that I think we really need to understand is these things, you get up, you open your window, you just gave up some of your privacy, because if somebody was watching you, they knew that you were at your place. It's just like when you get out of your house, you just like somebody can breach your privacy. It's just like whatever you do, whatever we do, whatever data we put out, it's going to have some sensitive information. It's about controlling the risk. So what we're trying to come up with here is, it's like we need to find objective measures. And NIH is actually very interested in that too. So we need to find objective measures to, estimate these risks and get it through to scientists and public. So because, you get consent from people, they give their data, but you actually did not really tell them what are the risks. Like you said, like, oh, there are all these initiatives, there's actually no like linkability study or something. That's exactly what we were thinking for the next step. I think, like, to be honest, we need a centralized way to measure these things. And we are doing open science, open data. That's good, but like you need to also give people a perspective about what could happen, what are the risks, and I told you guys there's 70% predictability. Well, what does that really mean? I mean, it's really hard to get these things into people's minds. But I mean, in general, medicine is like that. You tell a person you have 55% chance of developing schizophrenia. Well, okay, well that's, I don't know. I mean, what does that really mean, right? I mean, so, but there's big challenge, and I don't have the exact answer. So we need to work more on that. I guess that's my point.
So on the same topic, say I give my data to 23andMe, even if they don't get hacked, they're obviously not going to release all that data, but anything they do release is going to be giving up some of my privacy. So, your linkage study looks at like, okay, if we have all of this data available, what can we connect? But did you look at all what happens if they do a study and release some bits of information, and how that can affect an individual user's privacy?
We didn't do that. That's a good question. Well, firstly, I don't think 23andMe, I think 23andMe could release some data. I mean, and you just didn't read those user agreements well enough. Not you, but people don't. I mean, who reads those things? I mean, and I was actually at a conference a month ago. There were people from 23andMe, and they were thinking about how to open their data to researchers. So there is actually, they're working towards that, and actually they should, because that is very valuable data. There's so much to be learned from that. But like I said, I mean, going back, like they also, actually they should be doing this stuff, right? I mean, they should be presenting this stuff to us, because, I mean, it looks like they actually, I mean, I don't want to say they don't know what they're doing, but I think they're still not there. I mean, they know what they're doing with genotyping and stuff for sure, but it's just like we still don't understand the risk, and that's a big problem, because right now, U.S. government is trying to recruit 1 million people to get genotype, and that data is going to be open. 1 million people, every 300 person is going to be genotyped. I mean, what are the risks behind that? I mean, we don't know.
I have a question about the data getting more and more big, going into big data. I saw Pat coming by that we're going from a hypothesis-driven research to a data-driven research, which is, of course, great. But the risk with that is having big data, having a big N number, doing any kind of correlation or regression with that. You will find a correlation or regression. problem in doing that, or in astrophysics or in biology, is that we don't know, if you don't know the data, but also not our subject too much. If you have a certain protein, we don't know exactly what that protein is doing in context of the cell where it's in and the part of the cell body it's in. So what I see coming by a lot are these lists of genes, for example, proteins with a hit list of 20 significantly changed proteins or genes in a certain disease, most of the time schizophrenia or autism. And basically without a proper understanding how this would work in a network, and then they put it in a network, and the network is huge, and there's no understanding for how that might how that node is affecting another node far down the network at all. So how do you make sure that researchers are not only understanding the data they're using, but also the concept of the data, their hypothesis, instead of actually claiming that they have a significant result? Well, probably if you collect that data again with that n number, you find way different kind of correlations. So how do you make sure that the quality that's outputted of this data is an actual hypothesis, is an actual kind of answering to the question, instead of a data-driven approach that just give a nudge in a certain direction, which is probably wrong?
Who are you asking? To me?
Any, because both of the research topics are gathering big data.
Maybe I can comment. So in astrophysics, we do not only one experiment to collect big data. Often, I think the part of the question here is, how do we know that I think there's a complexity in the process of obtaining data, such as understanding telescope and how it collects photons, register, and every step of these processes matters. In this process of collecting data is getting ever more complicated. Not just the data is growing, but there's a complexity in the way you acquire the data. And one can never be sure that there's no flaws in the process of collecting data, and that creates I think creates the questions about the integrity of the data that's collected. Often what we do in astrophysics is to at least do 2 or multiple experiments of the same kind. And in fact, that's what's happening in this galaxy surveys. There are half-dozen galaxy surveys done by different countries. Instead of joining up and do one bigger experiment, we're keeping them, especially in astrophysics, separate, independent experiments, so that you can do cross validations of different results. And often the one team actually claims the discovery of dark energy, and the other team may actually falsify that. Oh no, but we didn't see it. So that process of checking and doing multiple experiments is especially important. This is what's done often, and without actually. So even within US, when you do things like cosmic CMB experiment, microwave band, the NSF is actually. It's important that agency funds not just one experiment or one that's better, but to actually think about supporting multiple experiments exactly for this purpose. And same goes for computational astrophysics. When you do experiment, we cannot just rely on one code that is thought to be superior, but you would always want to look for alternative. Otherwise, we would not know. Often we do not know the right answer because we're solving this non-linear, very complicated equation where we don't know the answers to, and that's what makes computation great, that we can tackle all of these questions that we didn't know how to solve. But then, because we don't know how to solve that one method, it is a chance that one method will not get that right answer, and only by trying something different that you would know whether you're converging on the answer. But there's even a question as to whether you're actually converging on that right answer. That's another deeper level of questions about whether you got the right answer.
Right, so no, I mean, in general, when you do association studies like GWAS, like genome-wide association studies, or these, I was talking about EQTLs, genotype to phenotype correlation rather than a binary, correlations. What people do is they have a discovery cohort and a validation cohort, And they so validate, like obviously discovery cohort is where you find these associations and you go and test. So there is actually a lot of statistical, I mean, there's tons of statistical methods and they are very strict on how they, how you find, a variant associating with a phenotype. And then on top of that, you go and validate it in the validation cohort. And then on top of that, people usually do animal models, using these, CRISPR, Cas9 assays. You go and knock down the gene, or you go and change the variants. Thanks to God, now we can do it, and then see if there's a corresponding change in phenotype. So there's actually very, strict way to validate it now. I mean, it wasn't there like 10 years ago, but now people do it because there was big problems with GWAS studies. Now they're much more strict. Well, I mean, this question actually relates a lot to this topic because, we are assuming these, EQTLs, that is the variants that are changing the expression levels or any phenotype is universally conserved in all people. And that's not true because, depending on the population, it's actually these, for example, in anything that works in a European population may not work but like any variant that changes expression in a European population may not change expression in a, African population. So, but actually in our study, I did a lot of tests like, cross comparison between populations and between different cohorts. It's pretty well conserved, so to speak. So, but that's a good question. I mean, these things are major challenges for discovery of these.
Yeah, just to follow up on the linkage risks. So given that you've shown that there are these almost kind of insurmountable or unforeseeable risks, that becomes a higher and higher hurdle for researchers to share data and make it completely anonymous and de-identified. So would you see it as we have to then accept some threshold of risk, so dependent on the data? So if, my user ID for Netflix then can be linked to me and people know that I rated Top Gun 5 stars, we say, who cares? Or versus, if my genotype can be linked to me and that has some risk if, say, employers or, health insurance agencies or government is using that information against me. So you have to really maintain a higher threshold there for acceptable risk. Or do we just have to accept that there's unforeseeable consequences and there needs to be like zero? That should be the goal, is zero risk.
Things. So firstly, in the Netflix case, nobody cares about you, you rated Top Gun 5. But what happens is, if you, for example, some people, there's a lot of stuff that people don't want to be revealed about their, movie choices. Not you, not me, but there are a lot of people who would care about that. So that is where actually, it's private. You don't want people going in there, You don't want people seeing a lot of like very political movies. You rated them 5 or 0, there's things, there are a lot of other things that could be assumed. People don't want, to be seen. You want to be left alone. You want it private, right? So, secondly, I think, yes, we have to accept that there's risk. There's always risk when we put data out there. But you have to, like in the consent forms or something, you have to firstly, like objectively and correctly predict it, estimate it, and transmit it to the public in an intuitive way. Those both of these things are very new. I mean, we don't know how to do it. There's this mathematical framework called differential privacy. It's the computer scientists love it. It's great, but if you apply it to the biological data, you just don't get anything out of it. It doesn't work. I mean, a lot of companies use it. Google uses it. Microsoft uses it. But if you apply it, people did that. If you apply it to the biological data, for example, the one paper was actually pretty cool. So they were using this differential privacy to, there's warfarin, this blood thinner drug. So it turns out, depending on how much warfarin the doctors assign you, you can go and identify the patient's genotype pretty certainly for a certain variant. And, you don't want that to be revealed out. And the paper basically used differential privacy to, set the warfarin dosages, but it turns out those dosages that were private were killing patients. So actually, you just lose all the utility of that data. So, there's now another, on top of that, there's another thing, it's biological utility. How do you define the utility of the data, right? So there are so many different factors that, we need to think about in this area, I guess. Does that answer?
So, Arif, it sounds like from what you're saying that the risk is in some sense time dependent. That the techniques that one has at your disposal to compare data sets or access different data sets changes over time. So is there an obligation on you as a researcher to update the consent forms for people who have given you samples and offer them the opportunity to redact their data from your data sets? And then going back to the open data issues that we've been discussing, does that then cause impact on data sets that may have been released openly in order to protect the rights and privacy of the individuals whose samples are involved?
That's a good question. Yeah, I mean, that is one of the things that are very vague, I guess. Not vague, but it's very hard to define. I mean, exactly. I mean, there's the time dependency. And actually, as a researcher, no, I don't want to write that in a consent form, but you have to, because then you would lose a lot of people. I mean, they wouldn't want to give their data, right? So, I mean, it's just we need, I mean, I don't know the answer to that. I mean, I guess it should be put there, but how it should be put there and what should be there, I cannot really say right now. Yeah.
Thank you. So both of your areas are heavily computational, and so there's a lot of use of software and code and computation. And I was at a conference a couple of years ago, and Tony Hay made a comment about how in the future PIs will be working with data scientists and software engineers, not as much as they work with grad students and postdocs now. So maybe if you can just comment a little bit on that aspect of your work, how much you rely on data science and computation, and how you see that, improve the work that you're doing and inform.
So yeah, over the last few years, we've had graduate students and postdocs who actually move into data science. So we are one of the producers of data scientists from physics and astronomy, because what we do is heavily computational, and skills you earn actually is helpful for the data science community. And at the same time, so we need to start to coordinate with the data scientists to help develop various aspects of the infrastructure that we need to actually do sciences as well. So I think this is starting to happen. That in the past, I think we have been sort of losing the people who are working in academia to the newly generated area of the data science. But then there's now also the convergence of working with the data scientists, and I think there has to be a partnership between the university and data scientists and possibly this industry to create the infrastructure. I think that there was not a sort of system to actually sustain the data scientists within academia, that there was it's a new position, and that there was, but I think it started to be realized that this is the very important part of actually getting the science done. So I think that the situation is gradually changing, that there are new data science institute which can host and develop the researchers, and then we have to work with the data scientists to actually develop software and also work on algorithm. I envision the data scientists are the sort of it's going to be a hub of different discipline, which can, I think data science can work not just with one discipline, but using some techniques and data and algorithm. If they work on different sub-disciplines, then these are the people who actually can connect and there's sort of the nexus of the knowledge transfer from one field to another. And I think this is a way forward in doing innovations because, for example, some of the techniques that we use in computational astrophysics came from engineering. And that happened 30 years ago, and we're still continue to use the knowledge and techniques that we inherited from other fields. And this has to happen at a much faster speed. And I think that successfully placing the data scientist at the nexus of the computational and data science research in the future, and we need to think very carefully about how to sort of make that happen.
Yeah, I agree with everything he said. I want to add just one thing. So, you know, well, I mean, the way I think about data science, I still don't have a real exact definition of it in my mind, but you know, like I would say like this totally kind of in parallel, but like 85% of the time that we are spending to, you know, do analysis is just gathering data, you know, getting rid of reformatting it, getting rid of that, you know, the noise, the stuff that you don't need, and getting to the final thing that it may be data structures, an Excel file, it may be something in the computer memory, but getting to the point where you can now apply some machine learning on it. But the problem is, genomics at least is going to a place where you just cannot fit something like, I mean, right now, you know, we had 1,000 Genomes project. You had, you know, like 3 million variants, 2,500 individuals, like, you know, gigabytes of memory, fine. But now we're going to, you know, like there's going to be 30,000 individuals, and then there's going to be 1 million individuals. There's just not enough memory. I mean, it just starts going into what he was kind of like, going to. It's going to a place where you have to do everything either like HPC level distributed, like how do you design those algorithms? You need a lot of computer scientists, data scientists, I guess, like, you know, for data wrangling, data, you know, cleaning and stuff. And you need people in biology that understand the output of those algorithms. You know, it's becoming a really like multidisciplinary, science. And I mean, I think the future is going to be we are going to do everything on the cloud. You know, because there's no way we can fit 1 million genomes, like in the whole raw data set on, you know, even on HPC. We just don't have that hardware, and we don't want to have that hardware because it's really hard to keep that up. I mean, we go back and forth with these guys every week. We piss them off, and they piss us off, you know. But I mean, it's just, you know, there's just, I think the way we have things is not very sustainable, and I think it's going to be very multidisciplinary in the future.
I have one more question. It's a question about the CRC. Very happy about that. And I'm a computational neuroscientist. I'm actually quite new here, hence my accent. And I looked through the site, and I signed up for a couple of things, waiting list. But what would really help me at the moment is to have someone or one-on-one or a group to discuss some models, some computational tactics, or some data structures. And I looked through the website and I couldn't find that. Maybe I overlooked that. But is there an initiative or people planning to do that as a kind of meetup? Okay, great. Great, thank you. That's all I want to know.
I'm so glad you brought that up, and I would absolutely encourage you to reach out to either Dr. Rob Bjornson or Dr. Andy Sherman regarding that. That's why they're here, and they love helping people out.